Thank you for tuning in to our Young Women in Bio virtual presentations. We are very excited to announce the availability of these online resources through our brand new YWIB online platform, which provides young women across the United States and in Canada free access to our educational and leadership programs and other opportunities in science, technology, engineering, and math. YWIB is launching this new initiative for young women who are curious about STEM and want to learn more, but may not live near one of our 13 Women in Bio chapters in cities across the United States and in Montreal. Our mission for Young Women in Bio is to give girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math. We strive to provide education and hands-on experience in STEM, and as well as share our passion for all scientific fields. The launch of YWIB Online comes at a time when there is a real need to provide girls who cannot otherwise attend our events and programs with access to online programming in STEM. This will help with our goal to ignite curiosity and to inspire young women to see a future for themselves in STEM. We believe that by making these online resources available to a broader audience of girls, we can encourage more young women to find a lifelong passion and career in STEM. YWIB is part of the Women in Bio with chapters nationwide and in Canada. Women in Bio is a nonprofit volunteer organization that promotes diversity and inclusion for women in the life sciences and beyond. We currently have 13 YWIB chapters across the United States and in Canada. To learn more, visit www.womeninbio.org/YWIB. Our two main speakers for this webinar are Dr. Stephanie Langel and Dr. Julie Steinbrink. Dr. Stephanie Langel graduated with her Ph.D. in Immunology and Virology from The Ohio State University and is currently a postdoctoral research fellow at Duke Human Vaccine Institute. Dr. Langel's research focus is understanding how the immune system responds to viral infections in pregnant women and children. Dr. Julie Steinbrink graduated with her medical degree from the University of Pittsburgh and completed her residency training in internal medicine at the University of Michigan. She is currently Chief Infectious Disease Fellow at the Duke University Medical Center. She specializes in infections in immunocompromised patients, such as solid organ and bone marrow transplant recipients. Her research focuses on genomic biomarkers of acute infection to improve diagnostics in this vulnerable population. Drs. Langel and Steinbrink will be sharing some of their knowledge regarding coronavirus biology and the clinical implications of COVID-19. Afterwards, Dr. Stephanie Ham, a YWIB volunteer and science director at Metabolon, will provide a summary of the COVID-19 research and clinical studies that is currently performed by companies in our area, the Research Triangle Park. Dr. Langel will speak first.
Great. Okay, so I've unmuted myself. I'm sharing my webcam, and I'm gonna go ahead and change presenters so that you all can see my screen. And we're going to get started. I'm really happy to be here. I want to thank the organizers firstly for inviting me to talk with you all today. It's really exciting to be able to share what I do with others, but of course it is kind of a scary time. There's a lot going on, a lot of uncertainty. Of course, all of you have had to stay home from school. So we are just appreciative that you're here to learn more about the coronavirus. And Julie and I are presenting kind of two different sides of this, this pandemic. And I'm presenting more of the virology side, and Julie present more of the clinical side. So I'm going to start by talking about what COVID is and some of the definitions to help you understand where these words are coming from. So COVID-19, what does that stand for? So it stands for Coronavirus Disease 2019. Breaking that down, the CO comes from corona, V comes from virus, the VI rather, and then the D comes from disease. And 2019 being that this virus was discovered and the outbreak was recorded originally by China and then the WHO in 2019. So what causes COVID? COVID is the disease that is caused by the virus severe acute respiratory syndrome coronavirus 2. And so you can see by the highlight or by the red bolded letters what that acronym means. So we have two different things: the disease, COVID-19, and the virus that causes the disease, SARS-CoV-2. If you want to think about it like HIV and AIDS, if you're familiar with that, HIV is the virus and AIDS is the immunocompromised disease that people progress to once they have the virus. So you could think of COVID and SARS-CoV-2 like that. But first, let's talk about what a virus is. So what I'm showing on the screen are many different types of pathogens that can cause disease or not in a particular individual. On the left are anything that is cellular, or what we would consider living, and on the right is anything acellular that we would not consider living. So it is interesting when we talk about viruses, because viruses fall under the acellular category. But when we describe viruses, we sometimes give them life-like functions, or we describe them as if they are living, because a lot of times it's easier to describe the pathogenesis or the progression of a disease by a viral infection by describing it as if it's alive. But by looking at this, we can clearly see that the difference between cellular and acellular is the ability of cellular to have things like a nucleus, to have all these different types of things inside the cell that do what? Produce their own metabolism. They are able to freely live in the environment, producing their own nutrients and living sustainably as a cellular organism. But viruses, on the other hand, they are metabolically inert, meaning that they cannot produce their own metabolites themselves, and they are particularly dependent on you or a host to replicate and produce copies of themselves. Without a host, without a cell, they will not survive out in the open, like let's say a parasite, protozoa, fungi, or prokaryote would. So we're going to move forward then thinking about the virus as an acellular sack of proteins that has to have a host to take advantage of inside of a cell. So what is a coronavirus? Well, let's first talk about the beginning of this word. The beginning of this word is Latin corona for crown. Now, or garland, but crown is kind of easier to think about. And I'm showing two different depictions of this virus. On the left is a computer-generated picture of SARS-CoV-2 that was helped by something called cryo-electron microscopy and computer algorithms to give this color and shape. If I look over towards the left-hand side, this is negative stain electron microscopy, and this is showing us a crude version of what the virus looks like, which is not as pretty to be able to show in different publications or on the news, but they're one and the same. And so a coronavirus, I'll talk a little bit about what you're looking at here, which is really the outside of the virus. But a coronavirus, like many other viruses, forms a family, which then can be divided into different categories of viruses. There's genus, species, subspecies. We're not really going to get into all of that because it would take too much time. There are many groups of scientists that decide taxonomically or categorically where do we put these viruses in terms of families, genus, species. But the coronaviruses make up a group of viruses that is really dependent on this outside crown. And these are called spike proteins, which I'll talk about in a bit. And you can see them protruding from the outside, and they play a huge role in the ability to infect you, but also to replicate themselves. So when we think about categorizing viruses, we are looking at the outside, but really what breaks them down is what is on the inside. So pretend you're looking at this very pretty computer-generated version of the SARS-CoV-2 virus, and you're gonna cut it, almost like pretend it's an avocado, and you're gonna cut it down the center to be able to look at this from inside the virus. And you can see there are many different types of proteins that make up this virus structure. But of course, this is a cartoon generated diagram, so we have to kind of, use our imagination, but you can see that the pink on the outside are the spike proteins, and those are represented on the as red spike proteins in the more pretty picture. But on the inside is where we can see the viral RNA. So this is an RNA virus, and if you learned about RNA and DNA within your classes, you know that RNA plays a particular function in the replication of a genome or of genetic content. And so it resides within this proteinaceous structure that is the envelope, the membrane, and then the nuclear capsid. And the goal of all these different proteins is to hold the structure so that the very important RNA that's wrapped in this nucleic capsid can stay safe. Of course, that's the virus's goal. We don't want it to stay safe. We want it to break apart so that we can try to neutralize the virus. But all of this is really what the virus depends on to be able to enter our bodies and replicate. Starting with the ability of these viruses to enter our bodies and be able to produce more copies of themselves, which is their goal. Of course, again, thinking these are not living organisms, but the main goal of this sack of proteins and RNA is just to keep making copies of themselves. And so the first thing that they do is something called receptor binding. Remember from the previous slide, the pink outside spike proteins are the first step to be able to bind to a human cell. And so the human cell is going to have, your cells are coated in proteins, many different types of proteins, not just the ACE2, which is what I'm showing here. But the virus is searching. It's searching for one of these receptors to be able to bind a hold of, kind of like a lock and key mechanism, to be able to for the virus to be able to then enter your cells through this receptor mechanism. Now, scientists have worked quite quickly to identify that the SARS-CoV-2 receptor is a protein called ACE2, or angiotensin converting enzyme 2. We won't get into the biology of ACE2, but just know that this is on the surface of your nasal epithelium and in your lungs, which we'll talk about in a bit. So that's step one. What I'm showing you now is, let's pretend that this is a cell, the human cell, and this is diagramming the outside, and you can see over here on the right-hand side, spike protein receptor binding. So that's what I just showed you back here, receptor binding, step one. And the cell has to be permeable, so once it binds, it's endocytosed, it's brought into the cell, and some of these terms you may have heard in science classes, but I won't go into too much detail because there are, this is a whole field, and people spend years, careers trying to figure out these life cycles of these viruses, because what Julie's going to talk about, they really matter in our ability to target different steps of this mechanism with drugs to be able to stop the virus from replicating in your cells and causing pandemonium of your immune response that's trying to figure out why is there a virus replicating in the cells. So you can see after endocytosis, it's brought into the cell, and the genomic content, which is depicted by these little squiggles, are going to come out and they're going to start replicating themselves. If we remember from earlier that viruses need a host cell to be able to replicate, it's basically using our own host cell machinery to be able to replicate itself. That is just rude. I think that viruses have been able to co-opt our cells by doing what they normally do to do what they want to do. And by doing that then causes us to get sick. So they're almost like little, very small parasites. And what you can see here is then after they make copies of themselves, they are coated back with those spike proteins, and then the arrow is showing on the left-hand side, they are released to then go infect another cell and to cause more problems. So now that you've learned a little bit about, okay, it has to have a receptor, the receptor has to enter the cell, it releases its genomic content, makes more copies of itself, and comes back out. Well, how does that work actually when you're standing next to an infected person? So why you hear a lot about wanting to stay 6 feet away is because scientists have discovered that this virus is spread by cough or sneezing droplets. Very small droplets that come from, after you sneeze, the mucus, and you can imagine when you sneeze, this is really what's happening, is you're having this kind of spray of things coming out. Now another person is going to potentially ingest that by sniffing by up their nose or in their mouth, and the virus can then start trying to find that ACE2 receptor, or the receptor to be able to enter the cell and start making more copies of itself. On the next slide, I'm showing that in more severe cases, the virus is moving from your nose and your oral cavity down to your lungs, where it really can start causing problems. So when we hear about majority of infections, 80% are mild, but there's about 20% that become more severe, and then almost 5% that could become so severe that a person could die. And Julie will talk more about that. But the virus, if it makes it down here, it's looking for more ACE2 receptors. And this is where the immune system starts to really react strongly to say there's a problem in the lungs and we need to clear this virus. And the virus is eventually, as it works its way, taking advantage of the alveoli of the lungs, which if you have been in physiology classes, you may know that's where the exchange of oxygen and air is happening. And once the immune system detects a virus replicating near these air sacs, it's going to start infiltrating cells that are good at killing viruses. But here's the thing, the viruses are inside your own cells. So your immune cells want to kill the virus-infected cells of the lung. The problem with that is it causes a lot of damage. And that damage then leads to something called edema. And the edema is where the alveoli of the air lungs start to fill up with fluid. Now, there are a lot of different labels on these air sacs. We're not going to worry about each one, but it's just to show that there are a lot of chemical interactions that are happening when the lung is detecting distress. Hence why it's called acute respiratory distress syndrome, because your lungs are basically trying to kill the infected cells, but they're causing a lot of problems along the way. In a person who's able to fight off this virus, maybe they did have ARDS, or acute respiratory distress syndrome, but then hopefully their immune cells calm down and the fluid decreases, and then they can try to go back to a repair stage of their lungs. But unfortunately, why people can die is because they can't get past this ARDS, where the lungs are filled with fluid and can lead to death. And so understanding that in terms of scientists' ability to prevent this from happening is really important. And so my last 5 minutes, I'm going to talk about, exactly where does SARS-CoV-2 come from? Because it's a question of, was this virus always around, and will there be more SARS-CoV-3, 4, 5 in the future? And viruses can come in many different forms in many different animals. Some viruses do not infect other animals. Poliovirus only infects humans. It doesn't affect other animals. But coronaviruses are known for their ability to jump host species. It's just the way that they've co-evolved with animals and us over many, many years. And they're able to do this because of the proteins on the outside, their RNA genomic content on the inside that we discussed. And what I'm showing on the right-hand side are multiple different genetically diverse, so the inside RNA content are different between all these coronaviruses. You'll find a similarity between where these originate from. Scientists have been able to determine that these viruses come from many times bats. And they do this by sampling the guano or the poop of bats in caves, and they find coronaviruses all over the place. But what's crazy is that the bats are not sick. The bats have evolved to be able to handle these coronaviruses and not be able to get sick, and they harbor them. And there are two others they're showing here that they think originate in rats, but you hear a lot about bats being the main reservoir. The thing with bats, though, is because they're able to harbor these viruses without getting sick, their body doesn't have any need to fight them off. They just get to symbiotically live with coronaviruses. The problem with that is as humans and people and other animals interact more and more over time, they can then adapt and change. And I say the word mutate, but that's not always a scary word. Viruses mutate all the time, but their ability to mutate over many years could lead them to infect something like a camel. And the camel can then replicate the virus and cause something called MERS, which is a different type of coronavirus that can cause disease. So what this slide is really demonstrating is that coronaviruses are going to be very hard, really impossible to get rid of, because they jump back and forth between animals and humans. And when we're specifically talking about SARS-CoV-2, we know from looking at the genetic content, remember inside of that virus structure of the RNA, we can sequence that with technology and be able to determine, okay, how similar is SARS-CoV-2 to every other coronavirus we've looked at? And luckily, scientists in the US and in China have looked at the coronaviruses in bats and being able to say, okay, this human virus is very similar to this coronavirus in bats. So we know that genetically they are linked. So we know that SARS-CoV-2 came from a reservoir host in the bat. What's really unknown right now, as I had shown in the previous slide, there typically is an intermediate host. We don't know what that is right now. There are some suggestions in the news about different animals, but it's really, we currently don't know what that is. And there's a lot of scientists looking to try to understand the transmission cycle of this coronavirus, because it's going to be important for controlling future transmissions. And so you can see on the cycle, once it goes into a human, and then what's different about this coronavirus from others, it has an ability to transfer easily to many different humans. So that is something in the bigger picture. So, we're focused on coronavirus, but we back up and we think about zoonotic diseases in general. We really are going to probably see more and more of these happening. And the reason is because the population of the world is growing quite a bit, and cities are expanding, and what that means for animals and wildlife is that we are becoming in more contact with them. The more interactions we have with them, the increased possibility that a bat, whose guano contains coronaviruses, can interact with a pig that's being farmed by somebody who is, growing their farm, or somebody who just has a cat, and urbanization is increasing their population of humans to where these animals are living. And this just increases the possibility of spread between animals and people. In addition, climate change really can make a difference here. If you have destruction of habitat, whether it be through climate change or just trying to extract natural resources for people living in cities to be able to live, like lumber, farming, you disrupt habitats that could then potentially contain animals that would spread diseases. So as we think about our approach to public health preparedness, there are many different ways that we have to tackle the ability to prevent something like this in the future. So of course, there's personal preparedness, which is your own ability to protect yourself by wearing masks, going to the doctor, making sure you're keeping up to date with your own health. But then in a pandemic like this, following instructions, like I mentioned, wearing masks, washing your hands a lot. The next one would be pandemic planning. And a lot of this is really dependent on governments and academic institutions, and also biopharmaceutical companies, to be able to understand what are the current disease threats and how can we solve that, whether it be through drugs or vaccine development. And that's going to require policy and partnerships amongst these different organizations that, innately as humans, we all have to work together, but can be, of course, challenging as we think about our own differences. But that's a really big part in public health. And then lastly, the response. And so what we're experiencing right now is, do we have enough personal protective equipment? How fast can we move to make sure we can all go back to work in a safe way? And all of these things, take funding to happen. And so governments, in terms of their public health preparedness, really investing in each of these 4 areas is really what helps to prevent things like the coronavirus pandemic from happening in the future. So we are actually going to be answering questions at the end. So if you have anything for me, just keep those in mind, because you'd have the opportunity to ask those at the end, or you can put them in the chat box. So as I end, I'm going to go ahead and transfer to Dr. Julie Steinbrink, who's going to talk a little bit more about the clinical aspects of the COVID or SARS-CoV-2.
All right, so hopefully you can all hear me okay. And hopefully you can also see my screen. It might take a couple of seconds to show up. Unfortunately, my webcam is not acting appropriately, but if you can still hear me, then we should be able to make it through that way. Looks like it might still be a white screen. Is that?
Yeah, I am, Julie, still seeing it as a white screen, but maybe we give it a couple more seconds, it'll pop up.
Yeah, it's a full screen on, there we go. That should be working now, correct? Oh, so it's working. You see what's happening is when it's like this, it's showing up, but when I try to make it as a presentation, it's showing up as the white screen. That's where my issue is coming up.
And I know, yeah, that happened before, but it seemingly had fixed itself within a couple minutes. But well, the organizers can also chime in, but I mean that's big enough.
Is that big enough to be able to see? Your screen sharing to show that it's showing your presentation slide, not your under show? There's different options of what you can show on the control panel.
The show. Let me try this. Is that better?
Yes.
Yeah. Okay. So you can hear and see me. That's all I need. Okay. Sorry about that. All right. So Stephanie did a terrific job about talking about the biology of the virus and how it replicates and what its host is. And so I'm going to be talking a little bit more on the clinical side because that's more what I see in the hospital. So first of all, well, what are the symptoms that you might see with coronavirus or with this particular coronavirus? And that's actually a tricky question to answer because it can be very different for people depending on who the person is that has the infection. The symptoms can be very diverse. They can be mild, they can be severe. They usually come on between 2 to 14 days about after you are exposed to the virus. So there might be some gap in time between when you were initially exposed and when you had your symptoms. And they can include things like fevers, chills, muscle aches, feeling like you got the flu. It could be a cough or trouble breathing if you get into that really bad lung injury that Stephanie had talked about. We call that ARDS, and those are the patients that oftentimes end up in the hospital. Sometimes people can have more minor symptoms like runny nose or sore throat. Some people will actually have GI symptoms, including diarrhea. And some people, interestingly, will also have a lack of smell and taste that comes as part of this infection. Although usually those, which is not commonly seen in many other viral infections and typically does get better as the infection resolves over time. So people of all ages can be at risk of getting in the infection, but who's the most at risk of having complications from the infection? The major people that we're concerned about are those older adults who are over the age of 65. And those can include people in nursing homes, oftentimes because they're older, they're in closer proximity to each other, so the infection can spread quickly. They oftentimes have other medical conditions that can make them more sick, and they have more difficulty caring for themselves. And people, even though who are younger but have other chronic medical conditions, are also at risk of major complications as well. And these chronic medical conditions can include diabetes, heart and lung disease like COPD or smoking or asthma, people with chronic kidney disease or need for dialysis or liver disease, people with severe obesity, as well as people who are immunocompromised, which means they have weakened immune systems, so like transplant patients, people on chronic steroids, people with poorly controlled HIV, and people who are on medications for certain types of autoimmune diseases like lupus or other things. So the spectrum can vary wildly, and it's important to know that you can potentially be spreading infection between people even before you're having symptoms, and some people don't even have symptoms at all. And so that's why it kind of leads into my next point, which talks about how can you protect yourself and others. Mainly as young individuals, you may not have some of the severe complications of infection, but there's still a potential that you could transmit it to others. And that's why we keep talking about recommendations from the CDC and other organizations saying, making sure to wash your hands very thoroughly for at least 20 seconds, especially if you've been in like a public place and touched a lot of surfaces, or after you've blown your nose or sneezed or coughed. If soap and water isn't readily available, you can use alcohol or hand sanitizer that contains at least 60% alcohol. Making sure to avoid touching your face, and in particular your eyes, nose, and mouth, because if the virus is on your hands, it kind of gets near your mouth. That can be a major problem. And in that instance, also making sure to clean and disinfect commonly used surfaces like your cell phones, your computers, your iPads, as well as doorknobs and things like that. You can use common household cleaners for that. Now, most of you hopefully have been social distancing, which means, keeping at least 6 feet away from other people outside of your home, because as we talked about, that's currently thought to be, how far the droplets can travel. But at the same time, sometimes you do have to go to public places like the grocery store for other necessities. And if you do that, making sure that you wear a mask whenever possible. Cloth masks are okay, and it's helpful for the community to have cloth masks, and then kind of preserve some of the other masks like the surgical masks and N95s for the healthcare workers when they need them. But also being aware that the masks are not necessarily a replacement for social distancing. They're kind of to use in addition to social distancing. And to also know that the mask is actually more so to prevent you from potentially giving the infection to another person, as opposed to them specifically transmitting it to you. But if we all wear the mask, then that reduces the risk of transmission. And like I said, even if you don't have symptoms, you may potentially still be a vector or a person who could transmit infection to somebody else. So how is this infection diagnosed? So that also varies a little bit depending on what hospital or facility or clinic you go to. The most common way that we test is through something called the nasopharyngeal swab. Naso meaning kind of you're going through the nose, but then you got to go way deep in the nose, as you can tell by the picture there, way to the kind of the back of the nasopharynx. And this test is the most easily accessible for clinicians to be able to do, in the clinic or through the drive-up centers, but also knowing that it has to be done right, because if it doesn't go deep enough, you might not get a good sample. And if it does go deep enough, it's going to be pretty uncomfortable. I can tell you from experience that patients will cough and sneeze and at you if you're doing it appropriately, because it triggers back there. So we do this because it's an easy area to be able to access as compared to other bodily fluids that we can try to get. In addition, with this virus, we've been able to see that they get decently high viral loads from this body location, and particularly early in the disease. And sometimes these tests can be positive for a long period of time, even weeks after you've had your symptoms as well. Although it's hard to know necessarily if that means that you're contagious or not, but we can still detect the virus for prolonged periods of time. Now, if the test is negative, this can mean a couple different things. Either you truly are negative and don't have the infection. Occasionally, you can have false negatives as well, which is either if the swab didn't go back deep enough and collect an appropriate area, or potentially if you got the infection really early in the infection, or if you caught it really late in the infection. This virus is kind of a descending infection, meaning that as the virus progresses and you become more critically ill, then the nasopharynx may have lower viral loads, but the airways may have larger viral loads. And you can detect those by sampling fluids in the lungs through something called a bronchoscopy, where they actually stick a scope into your lungs to be able to sample some fluid and send that off for certain kinds of culture as well. And sometimes that can be positive even if the swab in the nose is not. However, that's a much more invasive procedure, and so certainly it's not the first line thing that we go to, but it is something if you're really sick and in the hospital and we need to know. Sometimes we can also test stool, and stool can be positive, but even in patients who don't have GI symptoms with their infection. Although again, it's hard to know exactly what that means as far as contagiousness or other problems. And then I wanted to get a little bit into how is this infection treated? And so why is it so difficult to develop treatments for COVID-19 and actually for viral infections in particular? In infectious diseases, we really struggle a lot with virus infections, and the medications we have can certainly be improved. Antivirus drugs have to be able to target specific parts of the virus's life cycle that it's necessary for it to reproduce and replicate. But in addition, you got to be able to kill the virus without damaging or killing the cell that the virus is in. And viruses are really smart and adaptive, and they replicate and reproduce so quickly that they have plenty of opportunities to mutate or change as they go on, which can develop, and so sometimes they can develop resistance to certain medications or vaccines. So we have to keep that in mind. And right now, a lot of our data is also pretty limited. We only have small amounts of literature out there, although it's increasing every day. And so early on, we only had data based on very small amounts of patients. And so what we're trying to do now in the setting of a pandemic is, while people are working on brand new medications to try to treat this infection, we don't have the luxury of time to completely create new drugs here. And so what we've been trying to do is actually repurpose medications that were developed for other illnesses and other infections, and using those to be able to see if those work to treat this infection. And there are three main areas where the antivirus medications are likely to have an effect. One of those being inhibiting entry of the virus and blocking it from getting into the cell at all, avoiding that whole endocytosis process. Another is blocking replication of the virus to keep it from reproducing. And then we also look at how can we block inflammation caused by the virus. So not necessarily even targeting specifically the virus itself, but a lot of the excess inflammation that goes on with infection can cause some of the major damage, even more so than the virus. And so the majority of drugs that I'm going to talk about are part of ongoing clinical trials at my center at Duke and nationally as well. So the first one that I want to talk about is one that's gotten a lot of press. That's the hydroxychloroquine, sometimes the other word known for it is Plaquenil. And this has actually been used quite commonly in the prevention and treatment of malaria. It's also been used in certain autoimmune conditions like lupus or rheumatoid arthritis. And sometimes people also give it with azithromycin, which is actually an antibiotic, so not usually used for viruses, but it has some properties of reducing inflammation, and so they think that that might be helpful with this medication. And the way that they think it works with this SARS-CoV-2 is they think that it can work to inhibit the entry of the virus, so keep it, block its ability to be able to get into the cell, and also try to dampen or soften some of the immune and inflammation that goes on with the infection as well, and kind of keep that from replicating. There are currently lots of studies going on with this, both for treatment as well as prevention. There are studies going on even for healthcare workers who potentially have been exposed to the virus, studies looking at if this medication is effective in preventing infection in people who might be exposed. And there's some early amounts of small data out, but they're limited by only a small number of people being enrolled in them. And a lot of people have had to drop out of the study because of issues tolerating the medicine. And part of the problem with that is this drug, particularly if given with the azithromycin, can have lots of side effects on the heart and can actually cause some very dangerous abnormal heart rhythms. And so, especially in people who have chronic medical conditions, and like I said, those are the people who are getting this infection, that makes it very challenging to be able to give them this medication and be safe about it. Additionally, even if we can give them this medication and we find out that it's safe, we got to figure out, well, at what dose is it effective and safe for the patient, and how long do we treat them? Do they really need a long course? Maybe they only need a couple doses. And that's information that we really need good science and good research to really be able to tell. If we're only looking at small amounts of patients or just a few anecdotal people opinions, then that can also be a challenge. So that goes with the first mechanism that I was talking about blocking virus entry into the cell. The next two drugs I was going to talk about focus more on the replication or the reproducing of the virus. One of these is called lopinavir. It's part of a drug called Kaletra. You might have heard about this earlier in the COVID time. This is actually a drug that we have used in HIV management. It's what we call a protease inhibitor. Protease is an enzyme in the body that's important for the replication of the virus. And this medication blocks the action of that enzyme. And so that kind of stops the life cycle of the virus and keeps it from multiplying. And in labs, it's shown some in vitro activity against other coronaviruses, but it's not been shown to be as effective for the SARS-CoV-2. And in clinical trials as well for it, there has not really been a significant difference in the clinical improvement of patients that were on this or not. Additionally, a lot of people have tolerance issues with this medication as well. It actually causes a lot of diarrhea and stomach upset, and it interacts with a lot of medications. And like I said, these people have chronic medical conditions and they're on a lot of medications, and that turns into a big problem. So for this reason, we're not significantly studying this medication as much, at least, at a number of institutions. Another drug that's shown some promise is remdesivir. This also acts, to affect replication by blocking the RNA synthesis. But it was actually developed and kind of had some research and clinical use in the Ebola outbreak. And it's a medication that's given through an IV to patients. There are studies going on at different institutions in the area, as well as through the biopharmaceutical company Gilead. And as far as, an early data going out for that. Now, again, like every other medication, it has some kinds of side effects, including it can potentially cause some injury to the liver, and we're not able to use, it has to be given IV as well, and we're not able to use it in people with bad kidneys because it can accumulate and cause toxicity in those patients as well. The other drug that is a potential option here is tocilizumab. And so while the other medications I listed were effective in treating the virus itself, tocilizumab actually works to try to kind of fight the inflammation that's going on with the virus. So the inflammation is managed through different cytokines, including interleukin and other ones. And this one blocks a particular interleukin 6 and tries to kind of suppress the inflammation that goes on in the body from that effect, and kind of trying to reduce the people from progressing to the really bad lung disease if they get this infection. It's also been used, like all these other medications, has been previously used for an alternative purpose. It's used in some autoimmune conditions, as well as with certain types of chemotherapy, again, to try to suppress inflammation. And in some early studies has also shown some benefits in breathing function and time to resolution of fevers and discharge from the hospital, but again, studies are still ongoing. It's also hard because this medication, because it reduces inflammation, it also causes problems with weakening the immune system. And so it might potentially put people at risk for other kinds of secondary infections as well, that you have to monitor for them very closely. Other medications that might treat inflammation, you've probably heard of steroids, and there's been some mixed evidence about if we could just give steroids to these patients. It may be helpful for those who have already progressed to the lung disease. However, there's also some evidence and studies that it might actually prevent the virus from getting out of the body longer, and it again weakens your immune system and puts you at risk for other types of infections. So that's a problem as well. Lastly, I wanted to talk about a treatment that's been in the news a lot, and that's something called convalescent plasma. And these are big words talking about taking antibodies from patients who have had the infection and have recovered, and trying to use that to treat other people's infections. Antibodies are part of the immune system, and they work to fight the infection in a couple different ways. Sometimes they keep the virus, they block the virus from getting in and infecting the cell, and they can also kind of mark the virus for destruction by other cells as well. And they can also be helpful in vaccine development too. And so when people recover from COVID-19, their blood or their plasma has these antibodies to fight the infection. And we're still trying to figure out exactly the significance of that. But what these people can do is the same way you donate blood, you can also donate plasma. And then people can transfuse that plasma into another person who has the infection but is struggling a lot with it, with the hopes that maybe that antibody may be able to help them fight their infection better and reduce the length or the severity of their disease. This has been helpful and has some evidence in the other coronavirus infections like SARS or MERS, and it has shown some effectiveness as well in severe H1N1 flu epidemics. And so we're hopeful that it also has an effect in these in COVID-19 as well, although, like I said, data is still limited. So it's possible that some of these will be helpful. It's possible that none of these will be helpful. It's possible that new drugs will come out. It's possible that more than one drug may be a good choice for this infection, depending on what other medical conditions patients have, or what age they are, or how severe their infection is. But what's really important here is that we have really good science and studies and, great women scientists and male scientists that, like all of you included here, and really try to investigate and learn more about this virus and how to best treat it. And with that, I'm going to pass it over to our last speaker, to Stephanie again, and then talking a little bit more about some treatment and other studies going on in the RTP area.
Thanks, Julie. That was wonderful. Thank you so much for sharing. This is a great transition into what I'm going to be talking about. Let me just share my screen. All right. All right, so I think we're good. So thanks everyone for being here today. I really appreciate it. I'm learning a lot from Julie and Stephanie. So one thing we kind of wanted to highlight was everything going on in the Research Triangle part, because we actually have a lot of great resources, a lot of work happening here, which is really exciting, and happening specifically for COVID-19. And I split up into a few sections, and I'm just going to kind of briefly show them and highlight maybe a few of them because we want to leave a lot of time for questions. But the first slide I'm focusing in on is the treatment. And it's a really great transition from what Julie was talking about. A lot of companies and universities are looking at developing antibodies to target COVID-19, to boost the immune system of patients so that they can fight this disease. Grifols is a company that is actually doing what Julie was talking about with taking the antibodies from recovering patients and putting them into other patients that are unable to produce the antibodies to fight the disease, to see if that can actually serve as a treatment. And BioCris is a company doing also the repurposing of drugs. So as Julie mentioned, they're actually taking experimental yellow fever treatment and now trying to see if it's effective against COVID-19 because it's shown a lot of effectiveness against different viruses, including coronaviruses. The University of North Carolina Chapel Hill is also doing some really interesting research. They did a test with mice that were infected with coronavirus with a new drug and found that it reduced the lung damage. But what was really interesting is that this drug can be administered orally, so it could be taken as a pill. And so this has a lot of advantages because you could avoid the IV, and also it has advantages from a drug absorption perspective. So you can absorb it better and it can get to the lungs easier. And the other two companies are also doing some vaccine antibody work. Heat Biologics is looking at a specific protein called GP96. And this is on all human cells, and basically it has a huge immune response when it's activated. So their platform's actually looking into just continuously activating this protein so that it can actually boost the immune system of the patients and fight the disease. So Another important part of this whole process is the assays and the testing to not only know what if patients have COVID-19, but also how to look into these vaccines and antibodies if they're actually working in patients. So a bunch of different groups that have launched this research as well. Advanced Animal Diagnostics is probably my favorite from this slide because they took a test that was initially used to detect disease in cattle and now are adapting it to be used for humans. And so this is just a 2-minute blood test to get a snapshot of the immune system. So kind of just repurposing is a really innovative way. Q2 Solutions is also unique on this slide because they have created an assay that can test vaccine candidates. And so this is going to be critical for clinical trials, and for all of those previous drugs that are being developed, we're going to need to know if they're actually working in patients. So this is an assay, a quick assay that can do that. The other companies, Metabolon, Laboratory Corporation, Cellex, are basically developing tests to look at disease versus non-disease and detection, which we need more testing to be available so people can know if they have this. And Cellex is unique in that their test is giving results in a very quick time, about 2 to 3 minutes. Or I'm sorry, 15 to 20 minutes. And then the next part that is really also important are the supplies. So the medical supplies are limited through all of this COVID-19 pandemic. We've heard a lot about the masks and the need for ventilators. And so these two companies have really been innovative in providing solutions for these problems. That NC State has developed a unique material that they can produce up to 500,000 surgical masks per day. And this material has excellent filtering capabilities and also could potentially be reused with the proper washing. So it's a really unique material and perfect for this need. Another innovative company, Biomed Innovations, they actually had a core technology that was designed for prolonging organ life for transplants. And they found that they discovered from COVID-19 that their technology could also be used as a ventilator design because it can transfer of air and oxygen through its system just as well as it can with fluid. So they designed and pivoted their whole company to creating these ventilators at 1/5 of the cost to provide to hospitals. So really great and interesting work happening right here in the Research Triangle Park. And for me, you know, it's a great source of hope and just to see this work happening and be living in this area that's contributing. So it's really incredible. And with that, I know we've already had quite a few questions come in, and we want to make sure we have sufficient time for that. So please put your questions in the chat box, the question box on the side, and we are happy to answer those. You know, most of them are towards, I think, Julie and Stephanie. So if they want to pop back on to kind of start addressing those, please feel free.
Great. There was a question that came in through email before we started. Do you want me to kick off the questions by answering that one?
Yeah, that sounds great.
Okay, cool. So let's see here. I'm going to go ahead. The question was, are there, I actually don't have the question in front of me right now, so someone can pop on and read it. But let me go ahead and switch
it actually came up in the question box too. So it just was, you know, why viruses like the coronavirus, talking about the seasonality of viruses and why, and you know, think of seasonally like the flu, and what does that mean for things like coronavirus?
Sweet. Okay, great. So this is just a little, this is an extra slide that I didn't get a chance to include in the original presentation, but wanted to show you. There are other coronaviruses that can cause disease, and the common cold coronaviruses, they do not have fun names to say. It's a little bit hard to remember these, but these 4 cause mild symptoms. And so we really don't pay much attention to them. They're not a big focus in research. And Julie, you can comment as to whether you even detect these or test for these in the clinic.
We do, but not specifically by name. Like we know of like on my test, it just says coronavirus, like in general, but not specifically by these identities. So.
Okay. And so what we know about these coronaviruses is these actually do have seasonality. You tend to become more infected with these during the fall and winter seasons. I think that influenza, because of the severity, you just notice it a lot more. So the seasonality is more apparent, but these do have some seasonality, although we don't study these and research these a lot because they do cause mild infections. That seems to be the thing with infectious disease research. We tend to study things more when they cause human problems of severity, which makes sense. I did also want to touch on these other two coronaviruses that do cause severe disease. So that's SARS and MERS. And again, kind of brings it back to the point that we know these viruses transfer between animals. SARS was in 2003 and 2004. It came from bats to civet cats. And then MERS was first reported in Saudi Arabia and came from camels. So that was a great question. They do have some seasonality, but you're right, they are different from influenza, mostly because they're the genome inside that virus particle. Influenza has many different segments, which allows them to recombine with each other when they're trying to replicate themselves to create new strains. And so that's why every year you have to have a different virus, a different vaccine for a new strain of influenza, because their genome, the genomic content, is just set up differently than coronaviruses. So you're unlikely to see that level of strain variation between season to season because coronaviruses do not replicate in that way.
All right, there's a number of great questions coming in. There was one about COVID-19 survivors and antibodies and their role in vaccine development and if they could help with vaccine development. And so in short, yes, they have a role in vaccine development, but we're still kind of learning in general what these antibodies mean, as what the clinical significance of these antibodies are. I am not specifically a vaccine researcher, and I can't exactly go into detail about that. I don't know if Stephanie can, but they have a role, but we're still investigating exactly how much of a role.
Yeah, I'm sorry, I was switching back to make sure my screen wasn't sharing. Can you repeat that question, Julie? My apologies.
Oh yeah, it was just they heard about the antibodies and if these antibodies would be helpful for us developing a vaccine for COVID-19, and if so, how would we do that?
Wow, yeah, that's a great question. I mean, antibodies, there's two ways to think about it. You can develop an antibody purely in the lab, called recombinantly, and you can then, if it's an antibody that targets the virus, particularly that spike protein, and it can neutralize it, you can give patients a in-lab produced antibody. So that's one way. But the longer lasting way is to learn about the quality of the antibody from an immune response that would help inform you about vaccine development. So when you hear about people donating plasma, who have been infected, inside their plasma are going to be antibodies that were generated against the virus. We can study those antibodies and be able to tell, okay, do they bind the spike protein well? Can they neutralize the virus? Are they long lasting? And so those are things that are going on right now at Duke and UNC and within the research triangle at pharmaceutical companies. So yeah, kind of two ways to approach that.
I don't know if you want to talk about, someone asked about animals like dogs and cats hosting the virus. And that talks a bit about your zoonoses part.
Yeah, it's a good question. So from what we know, this virus does not infect dogs, but it has been shown in some small studies to infect feline or cat, within that species. And I think it's unknown if our cats can get sick. I think Julie can talk a little bit about some research, really cool research she's doing to figure that out. But it has been shown to be able to replicate, produce more copies of itself, the virus, in cats, which is why we heard a little bit about that tiger that tested positive at the zoo. I don't think that cats are a main mode of transmission. It may be that this virus kind of takes advantage of the fact that, oh, hey, we're being spread amongst domestic animals. We can bind this receptor in cats. It doesn't seem like cats are really dying, so I'm not predicting that cats are going to be a main way that this virus is transmitting, but we actually don't know a lot yet, so I think more research is needed. And Julie, you're just
actually, oh, sorry, we can go on
that, right?
The other question is how exactly does the virus manipulate the cell into replicating its RNA?
How the virus manipulates the cell to replicate RNA. That's a great question. So if you think back to that cross section of the virus where you got to see both the structural proteins, but inside is the RNA, and the RNA, when it's replicated, produces 16 other proteins that I didn't even go into because it would be a whole another lecture. But these non-structural proteins, they, along with the structural, help manipulate the cell to produce its own copies of its own genome. So we know a lot from other coronaviruses of what these non-structural proteins do, but it all comes down to what proteins are being translated from the genome of that virus. Those proteins manipulate the cell. And you'd be welcome to email me if you want to know, if you're really curious as to what exactly, there's 16 of them, so which one does what? I'd be happy to further answer that.
And there's one more question to follow up with the seasonality question. And so just a little deeper, someone asked if it's true that viruses will go away in the summer and if temperature will affect the virus.
Julie, I'll let you answer that and then I'll follow up with whatever is left.
Yeah, so that is a very challenging question. So some viruses, like influenza and things, you do see decreased prevalence during the summer months and things. It's unclear though that that's necessarily going to be the case with coronavirus. We're still learning specifically more about this because there are still areas throughout the United States that have warmer temperatures that are staying. That that the heat, that the virus has still been active there, and some of the other novel coronaviruses like MERS and SARS have been in areas of higher heat and have still been able to survive there as well. I know there are a number of studies going on about temperature and heat and how that affects it, and some of those are in more specific lab recreated scenarios. It's a little different in the general environment. Our hope is that there is some decreased prevalence in summer months and things, but that's also the time that people tend to gather together and become more social and things, and so there's also potential for transmission and other things for other reasons. So that's a challenging question to answer.
Yeah, that was great. I don't really have much follow-up other than to say what we know from flu we can't really apply to coronaviruses being different, and people are getting infected in areas of the world that are very hot. So, but we do know immune systems can work better when there's more humidity and a little bit of temperature. So I guess we will just unfortunately have to wait and see. I'm unfortunately not able to see the questions, so I think we'll have to read them.
Yeah, I can read.
One is why do people... Oh, go ahead, Julie. I'll let you go ahead and take that.
I was just reading the next one that said, why do people seem to respond so differently to the virus, and how do you think our genetics might be involved in that? And that's another multi-layered, terrific question, and a multi-layered one. It's actually one that different labs, including my own, are trying to kind of figure out. There's a number, so I mean, one, different hosts are different. Some people have, if you have underlying medical conditions, if you have immunosuppression, if you're on other medications, that can certainly put you at risk. And so that can put you at risk for major complications. If your immune system is already abnormal to begin with, and then you get an infection, that's a problem. Additionally, certainly, I think that there could be a role for genetics, and in particular, they're looking into things like how, specifically with the receptors and how the virus kind of attaches in your lungs and things like that. And that may be a factor. And so I certainly think that there is a role for genetics, but I think that it's that on top of other things going on, like with the environment and with other things going on with the patients.
Yeah, definitely. And I was just talking to somebody about this, and it might help you think about, how would you design a study to be able to test that question? Because surely genetics does play a role, in addition to things Julie mentioned. So what you could do is you could look at all of the cases and remove any severe cases of people who are in a high risk category. So that would be older individuals, people who have comorbidities. Comorbidity would be diabetes or high blood pressure, heart issues, lung issues. You remove all those people and you look at those who would not normally be suspected to get infections. So young people, or even people who are in their 50s and 50s who don't have other comorbidities. You can then sequence their genome and look for something called single nucleotide polymorphisms, or SNPs. And those SNPs are just differences within the genome that are subtle, but that could play a role in immune cell trafficking, which would be the ability of the immune cells to make it to the lung to fight the infection or produce a particular protein. And so that's the way you could design that study, is remove the confounding patients and just focus on those who would not normally be suspected to get sick, and then start looking at their genetic code to see what the differences are.
The next one that I saw asks what a ventilator does and how does that help fight the virus. And so I want to make a good point. It's a great question. I want to make a good point of clarification here. A ventilator is a specific type of breathing machine. So when someone has so much trouble breathing that they can't breathe on their own, we have to give them escalating amounts of oxygen and help them out. And so sometimes we can do those through just nasal cannulas, where we can just give them oxygen through the nose. Sometimes we can do a face mask, but sometimes they need so much help that you have to intubate them, which means putting a tube down into their lungs and making a machine kind of actually breathe for them. Now, what that does is that helps the body breathe on its own as they fight the infection, but the ventilator does not specifically fight the infection as well on its own. So that's a supportive measure to help the patient as they're trying to deal with the injury that's happening to their lungs, but that alone is not going to specifically attack the virus and kill the virus by itself. There's a question here. I'm not 100% sure if I understand. It says the masks have filters and face coverings don't. Why are they saying to use coverings? And I'm not sure if people are talking about like surgical masks and things versus cloth masks. I'm not exactly sure. Hopefully someone can clarify.
Yeah, I'm looking at, so I had to come into lab today, and we get screened when we come in, and we get a sticker signifying we got screened. You can see my sticker, and we have to wear a mask. If I took mine off just for this presentation, but so this mask, what you're seeing and maybe what they're talking about is that there is a filter covering, and maybe the homemade cloth ones wouldn't have that. But I think even with these, in addition to the homemade ones, they are not considered N95, so they actually can still filter, that you can still get sick from this. The main goal of these face masks, in addition to just facial coverings, is that if you're sick and you don't know it, which seems potentially is happening with this virus, you're not spreading it, you're containing your sneezes, you're containing everything. But it would be correct to say that an N95 or something that a hospital, like what Julie would be wearing maybe in the clinic, and she can speak to that, would prevent viruses from entering. But the goal of this really is just to decrease people from spreading it to others. You can't guarantee that you yourself are always protected, particularly if you're using it wrong. So like if I'm doing this and I'm pulling it down to talk to you, or I'm pulling it down to eat, it's not going to work if I'm putting my hands all over it. So there's definitely a right and a wrong way to wear them.
Yeah, that's exactly right. And so in general, cloth coverings are okay in the community. And surgical masks are okay. You know, even in the hospital, I wear surgical masks most of the time right now. But if I'm going to be in a specific room with a patient that I know has COVID-19, that's when you switch over, because you outside of the room and other things, you want to make sure that there's no potential asymptomatic or pre-symptomatic transmission where a healthcare worker may be spreading it to somebody else. But when I'm going into a room with someone who has COVID-19, that's when I switch out to the N95, which is the special mask that has a special kind of filter that's helpful to reduce the infection to me as well. The next question I see says, what was the drug that creates abnormal heart rhythms? And could this be why some people have been found dead by heart attacks with COVID? So the main drug that I was talking about is the hydroxychloroquine, and in particular, if it's given with azithromycin, because both of those can affect your heart rhythm, and in particular, a part of it called the QTC, which can cause arrhythmia problems. So certainly that can that can have some heart toxicity problems. However, you know, some patients there's also some thought that COVID-19 can cause some increased number of clots in bodies, and particularly with bad infection, and those clots can be in a number of different parts of the body. We've seen them in the skin, we've seen them in the lungs, and in the heart as well. So it's also possible, and strokes as well. And so it's possible that some of these people might be developing clots in response to their bad infection and sepsis. And so that may also be contributing to heart problems.
There's a question about, is the coronavirus beneficial to bats? And that's a great question. We really don't know a lot about bat immune systems. Obviously, they're very hard to study in a lab. There's maybe a handful of labs in the country that have bat colonies housed inside universities that would allow you to study that. It would suggest that the bats living symbiotically with coronaviruses in their gut, it could be beneficial to them. And that's actually that would be a question I would love to study. Interesting things about bats, their immune systems, they run metabolically at a lot higher speed than we do because they fly, and their body temperatures, I don't remember the exact number, but their body temperatures are a lot higher than ours. I think they maintain at like 105, 107 degrees. And so there is a thought that because they maintain a natural body temperature that is what a fever or what would kill us, is why these viruses don't cause problems, because they can't maybe replicate or cause infection because their body temperatures run so high. And bats produce these special proteins that are antiviral. So it's a fascinating question, and I don't think we know, but it's a potential that they're maybe beneficial.
Yes, I'm really impressed with these questions here. Another one I saw is, if older adults are most at risk for infection, how are the clinical trials of drugs tested on these patients or on those who are immunocompromised?
That's a great question.
That is an awesome question. And, I'm glad you guys are thinking, exactly like what Stephanie said, thinking about what, if you were to design a trial, how would you do it? And and the thing is, it's also very particular to the type of drugs. Some studies have what we call inclusion and exclusion criteria, meaning that there are certain things that people need to meet in order to qualify for the drug. Like maybe your infection has to be of a certain, you have to have severe infection, not mild infection. And then there's exclusion criteria, where some of them, where say we flat out can't include that patient in this study because we're concerned about toxicity or problems with the medication. So just age or just immunocompromised for many of these drugs may not necessarily be a blockage for them from getting on trials to study these drugs. It depends on what other medical conditions or what other things are going on. This might depend on, again, specifically what drug is being studied. Like I said, the remdesivir is not going to work so well if you've got bad kidney disease, and so that's an exclusion criteria. You can't go on the study for remdesivir if you have bad kidneys. But specifically just being immunocompromised may not necessarily be it. But it's just you have to be cautious and thoughtful with these studies, and and sometimes, some of these drugs are studied initially in patients who are younger and healthier, and then applied later to other populations, and then you see how they do after it's clinically applied as well. So, and that's what we call like post-marketing. So after the drugs are available, then seeing how they kind of apply to the population and continuing to see how people respond to them even after the initial studies are done.
That was a great answer that I wouldn't really have known. So thank you, Julie. Looking at these questions, there's a really good question and kind of a big one right now. And it's asking about the controversy surrounding coronaviruses being engineered in a lab, and if we can comment on that. So what we know from scientific evidence is that this virus was not engineered in a lab. The thought that it is a very political conversation. So I think when we talk about this pandemic, we're dealing with repercussions that have to do with economies and governments and politics. What I'm going to talk about is just the science. I'm not going to talk about the politics. When this first emerged, it was announced by the WHO on December 31st, 2019. The coronavirologists of the world, which I did study coronaviruses in my Ph.D., so I was on December 31st, like I was getting emails from my colleagues about this, like, okay, this is interesting. Is this another SARS that we saw in 2003? And we asked that question. We say, okay, if it's coming from another country, let's ask the question, the hypothesis-driven question. And that hypothesis would be either the virus was made in a lab, or the hypothesis the virus was not made in a lab. And the scientific way to approach that then is to test that hypothesis. And what scientists have done who work with recombinantly engineered viruses, and what this means is we all have the capability, the technology is there to build viruses by their pieces of RNA. We can sequence the RNA. We know the sequence of this virus, of influenza, and you can build the virus to learn about it, which these experiments are very important in the lab to try to understand, well, how do we stop the virus with this drug or this vaccine? And so scientists put it to the test. They said, looking at the sequence, could anyone in the US, a US scientist, could we build this virus? And after doing, it includes wet lab experiments, which means at the bench, you're culturing the virus in cells and you're growing it. So it includes wet lab. It includes computer algorithm-based science as well. And so that would be bioinformatics, which is definitely a field you should get into. And one thing that I wish I had more training on was bioinformatics, because there's a huge demand for you if you can become skilled in this area. But the marrying of those two, you can look at the sequence bioinformatically and wet lab. It'd say that the sequence of this virus is not something that could have ever been predicted outside of nature. It doesn't make sense, meaning that the virus is working in a way that we've never seen before, and our scientists have the same technology knowledge that scientists in China do. So it would be very surprising if they were able to figure this out, because the sequence is in a way that evolved from bats. And again, I go back to, we compare these things to other coronaviruses we've sequenced. We know that it's similar to bats, but that the genome, the predictions of how would you make this virus doesn't contain those predictions. So that's kind of the first thing. The second thing, how we make viruses in the lab is you basically have a backbone, and like a car, maybe you're adding on wheels and you're adding on different things, but there's always a backbone. This backbone, there's no known backbone to this virus that exists in terms of the sequence that people have looked at before. And so what all of this leads to say is that when you're trying, this would just not be a virus that somebody could create, that nature does a really excellent job at selecting for certain viruses that are primed to replicate in our own system. So outside of transmission in animals over many, many years, this virus could not have become the way it is by just making it in a lab. And so that scientifically, that hypothesis was then to say, if the hypothesis was this was made in the lab, it was disproven. And a lot of the talk about it is really people want to put blame on somebody for this happening. And it's a political conversation, not a scientific one. But it's a great question.
And Stephanie, kind of as a follow-up to that, the next question is, if this current strain does mutate, how do we think this is going to affect humans? Would it potentially be more serious?
Oh my God, I love this question because the mutation, so what you can think about viruses is they all mutate all the time. It's just like what they do. Viruses mutate. But coronaviruses actually mutate very slow. And the reason is because I actually have a chalkboard here, let's see, or a marker. If this is the genome, I'm just going to draw a straight line. And a mutation changes the genome at one nucleotide base, like let's say right here. This virus has an enzyme encoded in it that can fix that. It will actually go back, fix the mutation so that it can continue to replicate just like it was before. And so coronaviruses do not mutate like the flu. So I mentioned the flu, it has so many segments and it's creating new strains all the time. Coronaviruses have this corrective ability to fix their own genome when mutations happen. So it mutates very slowly, which means there are not going to be a lot of strains because a different strain is just mutations that accumulate over time. So I would expect, based on what we know about coronaviruses, that it's not going to mutate enough that we're going to have as many strains as, let's say, the flu. So hopefully one vaccine should do the trick.
Are we still doing okay with questions? Are we out of time?
We can go a little bit longer. That's fine.
Okay. The next question is, what specific place in the body do antibodies come from?
Oh, that's so good. Julie, you can take that one.
Yeah, so antibodies are produced mainly they're produced in the immune system predominantly by cells called plasma cells, and they're oftentimes secreted by cells called B cells. And so a number of cells of the immune system are involved in antibody production, but it's not necessarily only, of the organs, the spleen is the one that's predominantly involved in the immune system, although, the antibodies can kind of be produced throughout the body as well. So there's not one specific place that I would say.
There's a lot of good questions.
This is a good one. Oh, sorry, you go.
No, it's probably the same one I was looking at. You go ahead.
Why does the flu affect even healthy people multiple times over, and you have to get the shot every year? And COVID-19 so far seems to only be affecting older adults.
Yeah, a great question.
That's a great question
Yeah, like why, the question of why this virus doesn't affect kids, let's say, and influenza definitely affects kids, and it mostly affects older adults, is a question that we are trying to figure out right now. It has a lot to do with the immune response and the ability of the lung to clear the infection. Why you have to get multiple flu shots has to do with what I had talked about before, where flu has multiple gene segments that recombine every year. And so we are constantly chasing the next strain and having to change the vaccine every year. Coronaviruses do not have these segments, and there's not going to be new strains of coronavirus like influenza every year. So that's a difference between the two.
This one says, why do young, healthy people get severe cases occasionally, and why might that happen if they don't have underlying conditions? And so, again, that's something that we're looking into, and that might potentially tie into one of our answers to an earlier question where we talked about, yes, the environment and other medical conditions play a factor, but there's potentially other genetic factors and other things that we might not be aware of. And so it's possible, although still needs to be investigated, are some of these people have other underlying medical conditions or other reasons, or I mean, not, genetic conditions or other things that we're not aware of that might potentially put them at risk for infection. I don't know for sure if that's the case with these people, but that's something, you know, that we'd want to study.
I think we're gonna maybe have to wrap up questions. I think most of these we've addressed generally, but I did want to comment that one of the last questions down here mentioned there was discussion in the news, people can get reinfected after having the SARS-2. Why is that? And it's a great question because it actually has to do with the way that we test for the virus. And so when we test for the virus, it's something called RT-PCR, or reverse transcriptase polymerase chain reaction, which is kind of long, but basically it's a way to detect pieces of the virus that you are shedding from your body. Shedding being in your mucous membranes, when you're sneezing, in your stool, and on your body. So you collect a swab, as Julie had, that was a really great demonstration of how far they have to go. Sometimes though, maybe they're thinking maybe they could just do the nose, but that has to be fully tested. But you collect those, and then you're looking for pieces of the virus. The thing is, when you test, you have to have a threshold of high and low. So when you get a positive test result back, you don't know if how much virus you have is really high or really low. The lab will likely just tell you positive or negative. But as a scientist, I could pull the machine and I could say, okay, the number of this patient, the number of viral copies inside their back in their nasal cavity is high or low. So that's an issue. If somebody is coming back with a positive result, but it's a very low amount of virus, they could go back to the doctor, get a negative result, but because your lower limit threshold is set at a certain place, when you're starting to clear the virus, the pieces of RNA will waver, positive, negative, because it's so low and it's very hard to detect. So the sensitivity goes down. And so those people were not getting reinfected. What was happening is they were getting a negative result because they had very low levels, and they would come back and maybe have a positive result. So it has to do with the testing, but I think it's a good point to bring up that we don't know if every immune response and antibody is protective against reinfection. That's something we have to learn down the road. But what we can say is in the acute phase or in the short term, you're not going to get reinfected. It's usually a testing. Your levels are so low that it's wavering and the sensitivity is low, so it's difficult to tell. You know, and I don't know, Julie, do you give people that number, that viral copy number, or do you just give them yes or no?
So for clinical use, we don't give them specifically that number. For research purposes, we trend. I mean, we keep it for our own investigative purposes, but for clinical implications, it doesn't significantly make a difference, the exact number, if it's still being detected.
Right. Cool.
Okay, so I think that will wrap up our webinar. We want to thank everybody for coming today. We hope you all have a better understanding of COVID-19 and the associated virus. We would like to thank our speakers, Dr. Stephanie Langel and Dr. Julie Steinbrink, as well as our YWIB volunteer, Dr. Stephanie Ham, for taking time out today to talk to us about COVID-19. I learned a lot, so I hope all of you students learned a lot, and I'm very appreciative of their time talking today. So one last thing. We have to thank our national YWIB sponsors. A lot of our events that we do in person, we get donations from these companies. So if you guys had a good time today at the YWIB webinar, look forward to some in-person events when things settle down and we don't have to social distance as much. And we hope everybody stays safe. Thank you for joining us. Young Women in Bio RTP will be hosting another webinar on May 14th. From 2 to 3 p.m. We hope that some of you will consider attending this. During our next webinar, we'll be discussing clinical trials and how technology is revolutionizing how patient data is collected. And this is very important in the time of COVID-19. How do clinical trials keep happening if patients have to social distance? If you're interested in learning more about Young Women in Bio, there is a Facebook page for our RTP chapter that we keep up to date for all of our events that you can follow, or there's more information on our National Women in Bio website. Another thing that Young Women in Bio has launched recently is Young Women in Bio Online. There are a series of YouTube videos on a YouTube page that other chapters have developed that go in depth on some of the topics surrounding COVID-19. So if you want to learn a little bit more about COVID-19, there are some YouTube videos online at this link here at the bottom. So once again, thank you everybody for joining, and stay safe. Hope everybody's enjoying social distancing. Thank you everybody. Bye.